So for today, it's a pleasure to introduce to you Dr. Michael Pluess. He's a chartered psychologist and a senior lecturer in Queen Mary University, London. He's also a visiting lecturer at the London School of Economics. His background is he initially trained in chemistry and music, and spent several years working in the lab and on the stage before pursuing his current interests in psychology. He has a PhD in developmental psychology and has taken a number of positions around the world, visiting lecturer in Basel, for instance. He's received a number of awards, which I won't bother to go into because it will tread on his time as a speaker. It's a pleasure to have you with us this afternoon, and Dr. Pluess is going to talk to us on Is Happiness in Our Genes? Thank you.
Thank you very much. Well, thank you very much. It's a pleasure being here. It's always good coming up from London to Cambridge. So I've been asked to talk about happiness and genes, and that's what I'm going to do today. I'm a psychologist, as Keith just said, but I'm looking at psychological development across life, including genetic factors. It's very important to look at all the different factors, and I'll mention that very quickly. So before I start, I want to would like to make a pitch for a book. This is an edited volume on genetics of psychological well-being, summarizing all the research that we currently have on genetic factors and well-being, and that came out last year and published by Oxford University Press rather than Cambridge. But it's the as far as I know, it's the only book at the moment that summarizes all the research. It's a research book. It's not that easy to read, but if you're interested in the topic, then this book will give you the background in behavior genetics, background in molecular genetics, and all the things that I'm going to mention today are mentioned in that book as well. So what I'd like to do today is I first want to talk a little bit about the determinants of well-being, because when we talk about well-being, we have to be aware that it's not just genetic factors that have an impact on our well-being, but there are lots of different factors, and we have to be aware that it's very complex. And then I'd like to present some findings based on twin studies. So these are the traditional heritability studies, behavior genetics. Then I want to look at some molecular genetic studies. So these are more recent studies when we are actually able to look at the DNA. So I'll present the most recent findings on that. And then actually my main research interest and my main contribution to the field is looking at gene-environment interaction, the interplay between genetic factors and environmental factors in the prediction of well-being. And that leads us to the topic of epigenetics, which I will just mention very shortly, because there is not very much research on that yet in relation to well-being, and it would be also quite complicated, but I have to mention it because it's very important to know. And then at the end, I'll conclude based on all of the research that we have, whether happiness is a function of our genes or not, and to what degree. So when we look at determinants of well-being, we have to be aware that the well-being that we feel today is the function of many different factors. So work, cognition, our temperament, personality, things that we experience, the prenatal environment as well. Faith and religion is a consistent predictor of well-being, predicts across all the studies that I'm aware of, it's the most consistent predictor of well-being. And genetic factors are also a potential predictor of well-being. But it's important to realize that genetic factors are just one of many different factors that have an impact on well-being. And that's true for any outcome that we look at, any behavioral outcome that we look at. Genetic factors are usually playing a role, but they just play one role, and there are many other players as well. And also very important to realize is that the genetic factors actually have an impact on many of those more environmental factors. So for example, there's research suggesting that the life events that we experience, these could be negative life events, stressful life events, things that seem to be independent of ourselves, they are actually also associated with our personality. based on our genetic predisposition, we are more or less likely to experience certain experiences. So genetic factors are associated with life events, with health, intelligence, our personality, our temperament, also the parenting that we receive. You know, that there's some research suggesting that children that are maybe a bit more aggressive due to genetic factors, they will evoke different parenting from their parents. So we know that the genetic factors have an impact on many environmental factors as well. Nevertheless, most people compare genetic factors and environmental factors and just treat them as two independent influences on developmental outcomes, including well-being. So I will have to make that point that that's not really how biology works, that's not really how life works, it's not just genes and environment. I'll talk more about that when we mention gene-environment interplay. But most of the research that I'm going to introduce now, so the behavioral genetic studies and molecular genetic studies, are actually based on this understanding that we have genetic factors and environmental factors, which is inherently flawed. But still, that research gives us some indication of whether there is some genetic component of well-being. So this is usually, genes are usually referred as nature and the environment as nurture, and a lot of people talk about the nature-nurture debate. From a scientific point of view, we know today that it doesn't really make sense to think in those terms. Is nature more important or nurture more important? We know that both nurture and nature is important. We know that both are intertwined, highly complex, in a complex way interacting with each other. So it's not just one or the other. There is certain traits that may have a stronger influence of the environment, and other traits are more determined by genetic factors. So the balance might be slightly different. But I'll come back to that later. So behavior genetics. Behavior genetics is the traditional way that researchers, scientists have used to get an understanding of how much of a certain trait is determined by genetic factors or by environmental factors. And the classic methodology is the twin study design. So twin studies have been used, are still being used to get an understanding of how much of a observable behavior, measurable behavior, is determined by heritable factors versus environmental factors. And the way this works is that in this classic design, we compare identical with fraternal twins. So identical twins, they share 100% of their genetic information. They're genetically identical. And fraternal twins, they're like any other siblings. They share on average about 50% of their genetic information. So if you have an identical twin sibling, you would have the identical genetic information like your sibling. If you have any other sibling, or if you have a fraternal twin, you would share on average about 50%, which is the same amount that you would share with your parents or with your children. So the idea is that you have those different twins. They differ in the genetic material, genetic similarity, but they grow up in the same environment. So if you have an identical twin pair, they grow up in the same family. If you have a fraternal twin pair, they also grow up in the same family. So what we can do in the twin designs is we can look at how similar are the two siblings to each other. And then we compare identical twins with fraternal twins. And because they both share the same family, the same environment, differences in their similarities must be due to genetic factors, right? So we have identical twins. If they are more similar to each other than fraternal twins, then we can conclude from that that there must be a genetic component that explains that they're more similar to each other. Does that make sense? All right. So that's the twin study design. It doesn't measure any genetic factors per se. It doesn't look at the DNA. It just looks at similarities and then puts that in relation to the genetic similarity between individuals. So behavioral genetic studies are not just, you can run those studies not just with twins, but with anyone that is related to each other. So there's other designs like the children of twins, but you can also look at adoptive studies. All these studies are behavioral genetics because they look at behaviors. They don't look at the molecular structure of genes. So they don't actually measure genes. They divide the variance of people into environmentally and genetically determined components. So there are two recent meta-analyses. Most of the research looking at genetics of well-being in this field is based on twin studies. And there are two recent meta-analyses. A meta-analysis is summarizing all the research that we have and combining them and seeing whether in combination they suggest one or the other, what kind of findings they suggest. So the first study is by Bangnes and Roisen. This is in one of the chapters in the book I just mentioned. So they looked at different outcomes related to subjective well-being, including life satisfaction. How satisfied are you with your life on a scale from 1 to 10? That's usually how it's being assessed. And positive affect. The sample included 15 different studies with about a total of 30,000 people. And what they found is that about 40% of the differences between people are accounted for by heritable factors, and the rest, about 60%, by non-shared environmental factors. So about 40% of the differences in well-being between people is determined by heritability, by genetic factors. So there is a genetic component, according to this study. The other study by Micah Bartels, as well published last year, looked at subjective well-being and life satisfaction as well, included many of the same studies, but an additional study which increases sample size substantially. And she found that the heritability accounts for 36% of the variance in subjective well-being and for 32% in the variance in life satisfaction. So slightly lower estimates, but still similar findings. There's always an error variance between those estimates. But in combination, we can say the behavioral genetic studies in total, when we summarize them on average, they seem to suggest that about 40% of well-being is determined by heritable factors, genetic factors. They range between 20 and 50%. There's looking not at just at happiness, but loads of different concepts associated with well-being or positive psychology, including things like spirituality, perceived meaning in life and purpose. All of those have been investigated as well, and they seem to have a heritable component. So one question and one interesting observation that has come up several times is that we can look at different constructs that are associated with well-being. For example, vitality. How much energy do you have? Life satisfaction. How satisfied are you with your life? How much positive emotions do you have? Things like that. How socially outgoing are you? And all of those things. So they are all constructs associated with well-being. Now, what we can do with those kind of studies is we can estimate the heritability for each of those different behaviors or constructs, and then we can see whether they overlap. So are the genetic factors predicting life satisfaction the same factors that predict vitality? In those kind of analyses, we can estimate that overlap. And what has been found that there's a large overlap between the different heritabilities of these constructs. So for example, one study looking at sociability and positive emotionality found that there is actually a genetic overlap, that the same genetic factors predict sociability and positive emotionality. And that has as well been found with vitality. How much energy do you have? So these are three different constructs, three different behaviors that we can measure with questionnaires and so forth. And they suggest that they all have an overlapping genetic component. So the same genetic factors seem to predict differences in all of these three different concepts. And this suggests that rather than having different genes for different well-being associated constructs, we do have a general positive genetic component, a factor of well-being that is reflected in lots of different well-being correlates or constructs. Does that make sense? So rather than having certain genes predicting one life satisfaction, other genetic factors predicting this outcome, it seems that there is one genetic component of well-being that's reflected in lots of different concepts. So that's an interesting observation. So the next question that people have been looking at is, we know that psychopathology, so the opposite of doing well, has also a genetic component, is heritable. So whether you're depressed, whether you experience anxiety, and so forth, that has a genetic component as well. So if you have a family history of lots of mental health problems, that increases the risk to have mental health problems. And that's the family history, and that's obviously for a big part genetic. So the question is, could well-being, the heritability of well-being, just be the opposite of the heritability of psychopathology? Or is there actually a heritability component that is unique to well-being? If it was just the opposite of psychopathology, then all we have is really a genetic component of mental health, and it's one continuum, and if you have a lot of psychopathology genes, then you're more likely to have problems. If you have fewer of them, you're less likely to have problems, and you have higher well-being. So that would mean that there is only one genetic component. But what we found, or what people have found looking at these studies of the overlap between well-being and psychopathology, they realized that there is a substantial overlap of about 40 to 50%. So 40 to 50% of the heritability of well-being overlaps with the heritability of psychopathology. But that still means that we have about 50% of the heritability that is not related to psychopathology. So we have a unique heritable component of well-being. We have a unique contribution, genetic contribution to well-being that is independent of psychopathology. It's not just the opposite of psychopathology, it's unique to well-being. So if I summarize that, the twin study findings suggest that about 20 to 50% of the variance of well-being is explained by a heritable component. And about 50% of that heritability is independent of the heritability of psychopathology, and that means we do have a specific genetic component of well-being. But what we don't know yet with these studies is, what are the actual genes that make up that heritability? All that we know here is, if you're related to someone, then you're more likely to be like them as well. And that predicts differences in well-being. But it doesn't tell us anything about what are the specific genes involved in well-being. So in order to find out what genes might be involved, we have to look at the molecular genetics. So now it becomes a bit more biological, but I hope it's still fairly easy to understand. So when we look at molecular genetics, we look at variations in the DNA. The DNA is a code made up of 4 bases here, and they come in pairs, A and T and G and C. We all have more or less the same genes. So it's not that some of us have one gene that someone else doesn't have. We have the same genes, but we have different versions of those genes. So we're not talking about some people having a gene, some people not having a gene. That does exist, but that's not what we look at when we look at genetic differences in people predicting common behaviors, common outcomes. So what we have is polymorphisms. That means different forms of the same gene. And we can look at those different forms of the same gene to see whether these different forms are differentially associated with a certain outcome. So a gene is a specific section in the DNA, and the gene would code for a specific protein. It's the plan to build a protein, and that protein then has a function, some function in the physiological system. So let's say this would be a gene. It's not that some of us don't have that gene, but some of us may have slightly different code in that gene. So I try to illustrate that here, right? So this is a C and a G, and some people may have the same gene, but instead of that CG, they have an A and a T here. So that is a so-called a SNP, a single nucleotide polymorphism, where the gene is exactly the same, but just one of those letters is different. And a gene is usually made up of thousands or hundreds of thousands of those different base pairs. So it's tiny differences, and there are millions of those differences across the genome, and that's how we try to understand genetic factors and the role of genetic factors regarding psychological outcomes, but also regarding lots of health outcomes. So we look at tiny genetic variations and whether they predict outcomes. And I'd just like to summarize some of those studies. These are all different methodologies to look at molecular genetics. So the first approach was to look at just the single genetic variants. That's called candidate gene studies. So some people thought that this particular gene must play a role in a certain biological pathway. So let's look at the variations in that particular gene. That's a candidate gene. There is a biological hypothesis behind it. And applying those methodologies, there haven't been many successful findings. So there have been some initial findings, but it was impossible to replicate them. So we don't really have a lot of single genes that are associated with well-being or really any other outcomes, to be fair. Then the more recent development is the genome-wide association study approach. Now, genome-wide association studies, they look at the whole of the genetic variation across the genome, across all the genes, usually looking at from 500,000 to 2 million different genetic variants across the genome, spread all across the genome. And these studies, they don't have a hypothesis of what specific gene would predict well-being, but they just test each single association of those millions of different gene variants with a specific outcome, here with well-being. And then they test, they correct for multiple testing, and this way they try to identify specific genetic factors associated with well-being. So it's a hypothesis-free approach. It's basically data mining in the genome to identify some associations. But this data is quite interesting because there are different things that we can do with it. It's not just that we can look at the associations, but it's also possible to calculate the heritability. The twin study designs, which I spoke about earlier, they rely on people differing in heritability. But with the GWAS studies, the genome-wide association study data, we can look at heritability between unrelated people, because we can look at how similar are they looking at those gene variants, and then we can look at how similar are they in their well-being, and then we can relate the similarity in the genetic factors with the similarity in well-being, and that gives us an indication of the heritability estimated based on common genetic variants across the genome. So this has been done in 2013 in a first study looking at 11,000 people, and they found that happiness and life satisfaction, the differences between people, about 12 to 18% of the differences between people can be explained by those genetic factors, looking at structural differences in the DNA. And we've run a similar study, which is not published yet, and we looked at the evaluative well-being, so that's life satisfaction, and we found actually a genetic contribution of 23%. And that's about half of the heritability that we found with behavior genetic studies, but that's usually what we do find, that with genetic methodology, we are only able to account for about half of the heritability that has been estimated with behavioral genetic studies. That has to do with that behavioral genetics includes more than just those genetic differences that we've been looking at in this study. The only study that tried to identify specific gene variants associated with well-being was published earlier this year, and it actually had a massive sample of almost 300,000 people. Now, what has been found that in the early days of these studies is that it was impossible to identify gene variants. That's the same, not just for well-being, that's the same for in medicine or any other research field that applies genetically informed methodologies. They generally seem to find that the identified genes can't be replicated. That all changed once people moved to very large samples. So with those kind of samples of 300,000, they have the statistical power to detect more reliably genetic factors associated with specific outcomes. So in this study, they were able to successfully identify 3 gene variants, but they had very small effects. So individually they explained about 0.01% of the variance in total. Those 3 different gene variants, actually all the genes involved in that study combined did not explain more than 1% of the variance. So that's a very important finding, maybe slightly disappointing finding, given that they had such a huge sample of 300,000 people and they were only able to identify 3 different gene variants out of millions of them that seem to have very small effects in the prediction of well-being. But this tells us something very important. It tells us that there is not a single gene predicting well-being. So that the 20% here found in these studies or the 40% found in the behavioral genetic studies are not the result of just a few genes. So it's not that some of us have a happiness gene and that's why they're happy. But we have lots of different genes that contribute to well-being. But as I mentioned before, looking at the behavioral genetic studies, which have about 40% heritability, now 20% of that is shared with psychopathology, there's only 20% that is specific to well-being. So the heritability of well-being is about 20%, probably half of that is explained by specific gene variants, those single nucleotide polymorphisms. So that suggests that our well-being has a genetic component, but the larger part of that is other factors. So summarizing the molecular genetic findings, we have to acknowledge that most genetic studies so far failed to identify gene variants of, with large effects that would predict well-being. Most small-scale gene behavior association findings failed consistent replication because many of these findings would be false positive findings. They're underpowered studies. One study finds an association, someone else tries to replicate it, they don't find the same thing. It's even more complicated because obviously there are ethnic differences in genetic factors. So most of the studies that people conduct are conducted with ethnically homogeneous samples, some usually Caucasian samples in the Western world. It's very different. The frequencies of these genetic differences are different across ethnicities. So if we find one finding in one ethnicity, we may not be able to replicate it in another sample because it's just different. So verified and replicated gene variants account for only a small fraction of total variation. So those 3 genes that have been identified, they account for less than 1% of the variance, and that suggests that the variability in well-being must be the result of many gene variants of very small effects rather than a few of large effects. So the genetic contribution to well-being is most likely the sum of thousands, of many thousands of different gene variants with tiny effects that in sum then make up the 20% that predicts well-being. It's not the case that we have a few genes that explain a lot of the variance in well-being. Does that make sense? All right. So that was the methodology looking at genetic factors and environmental factors and trying to understand how much is predicted by genetic factors, how much is predicted by environmental factors. But that does not really reflect the biological reality. So much of the research tries to understand how much is genetic, how much is determined by the environment. I think that's the question that most of us have. We want to understand how much of our well-being is actually determined by genetic factors because we think that if something is determined by genes, we can't influence it, but we can influence it if it's environmentally determined. So these studies, as I mentioned before, looking at well-being, they usually find that 40% of differences in well-being are predicted by genetic factors and 60% by environmental factors. But this model is not correct because it suggests that that there are independent, discrete influences on well-being, that a specific component is genetic and a specific component is environmental. And that's a misconception that is very difficult to get rid of because of a particular a paper that suggested, although the author themselves didn't suggest that, the people reading it usually understand it that way. Let's say that about 50% of our well-being is determined by genetic factors, and you can't change any of that. But about 10% of the other 50% really are determined by environmental factors, and you can change those. So then you can influence your own well-being. But this is suggesting that an individual's well-being, so your well-being, would be the result of 50% of genetic factors and 50% of environmental factors. And that's what's not true. So all genetic studies, the behavior, the twin study designs, but also the molecular genetic findings studies, they look at differences between people. They don't look at the individual. So some misconceptions here. So behavioral genetic findings, but also molecular genetic findings, they always refer to differences between people of a particular population. They can't be interpreted on an individual level. So we measure well-being in lots of different people, and that gives us a variance. So some people have low well-being, some people have high well-being. That difference is what we explain with those methodologies. So we can say that 40% of the differences in well-being between people is explained by genetic factors. But it doesn't suggest that an individual's well-being is explained 50% or 40% by genetic factors. Also, behavioral genetic findings are correlational. They do not inform us about causality. They don't tell us that genetic factors predict in a causal way well-being. They just say it's correlational, right? Very important. And as I mentioned before, behavioral genetic findings, they imply a false dichotomy between genetic and environmental factors. And that's because these methods are based on statistical considerations rather than biological considerations. Biology doesn't work that way. It doesn't work like you have genetic factors and then you have environmental factors. Biology is always both together. So we have to kind of forget that idea of that we can differentiate between genetic and environmental factors, and need to understand that they're always both. Genetic factors will influence environmental factors. Environmental factors will influence genetic factors. So we have gene-environment interplay. That's how biology works. That's how genes work. Genes create are the building plan for proteins. Proteins interact with environment. So there's two different kinds of gene-environment interplay: gene-environment correlation and gene-environment interaction. I will talk more about gene-environment interaction, but what I mentioned early on when I talked about the determinants of well-being, that genetic factors predict the life events that we experience, that's gene-environment correlation. So for example, you have a happy person. That person is happy due to genetic factors. That means that person is more likely to spend time with other happy people, or with environments that are selected by that person. So the environment someone experiences is not independent of the genetic predisposition, but the genetic predisposition increases the likelihood, the probability of experiencing specific environments. And the same way, someone who's very socially outgoing, if they would have children, they pass on their genes, but they also pass on the environment. So socially outgoing parents have lots of friends over, seem to be very positive and so forth. They will have children that are more likely to have the same temperament personality, but they also inherit an environment that is more socially outgoing and more reflecting that positivity. So genes and environment are usually not independent, but they're correlated. That's also true for if you're more miserable, you're more likely to evoke that reaction from your environment, right? Or as a child that is smiling will evoke more positive interactions from other people. That's a reality. So there's gene-environment correlation, and then there is gene-environment interaction. And gene-environment interaction means that we may have an environmental effect on well-being, but that relationship, that effect, might be moderated by a genetic factor. So if you have one particular gene variant, that relationship might be very strong. If you have another genetic variant, that relationship may be very weak or may not even be there at all. So our genetic predispositions can moderate the effect that environmental factors have on our well-being. And I'll talk more about that. So to summarize that, we know that there are genetic main effects and there are environmental main effects on our well-being. We know that we know based on experimental studies. We know that there are causal impacts, causal influences of both genetic factors and environmental factors. But we also know that there is gene-environment correlation. So genetic factors are not independent of environmental factors. And we know that there's gene-environment correlation with genetic factors moderating the relationship between environment and well-being, and environmental factors moderating the effect that genes have on well-being. That's the same thing, but just from a different perspective. So I would like to talk a little bit about gene-environment interaction regarding well-being. And that's where I'd like to talk about a specific gene. That's the serotonin transporter gene. The serotonin transporter gene is on chromosome 17. So that's an illustration of the gene. And if that gene is transcribed and translated into a protein, it ends up being the serotonin transporter. This is a neuron. So this is, let's say, in the brain. That neuron releases neurotransmitter serotonin. That serotonin is being released in the synaptic cleft and passes on the signal to the next neuron. So that's what happens in the brain. So then the serotonin is released. In order to be used again, the serotonin transporter, that protein, takes that serotonin has been released and transported back into the neuron so it can be used again. It's a recycling mechanism. So that's the serotonin transporter. So we know exactly what the gene does. If that gene codes for that protein, and we all have that. But there is a polymorphism. It's a slightly different polymorphism that I showed you before. It comes in two variations. So some people have a short version in the promoter region of the gene, and some people have a long version. Now, if you have the short version, you will produce fewer of those proteins. If you have a long version, you produce more of that protein. And having more or less of that protein has an impact on the serotonergic neurotransmitter system. So if you have fewer of those transporter molecules, the system is not as responsive because the serotonin will linger a longer time in the synaptic cleft. If it's removed quicker, then the neuron can fire again more quickly and is more responsive. Does that make sense? Okay. So we all have the serotonin transporter gene, but some have the long version and some have the short version of the same gene, right? But we all have two versions of the same gene, except men on the chromosome 23, right? So we have genes from our father and from our mother. They're being mixed up. And then we each have two copies of the serotonin transporter gene. That means there are three possible combinations. Some people will have two long versions. Some people will have one long and one short version. And some people will have two short versions, right? So there are three different combinations. There are two different genetic variants. And because we all have two copies of the same gene, we have two complete sets in our DNA, we can have either one of those three different combinations. And this is how we do research in genetics, trying to understand, are these different combinations differentially associated with a specific outcome? Is, for example, that one more associated with a disease or with depression and so forth? That's how we usually do. We have those three groups. We compare these three groups. So it's not very complicated. It's just looking at three different groups. So this gene is one that has received a lot of attention in psychology and psychiatry because of this study. In this study, they looked at maltreatment in childhood and whether childhood maltreatment would predict the probability of developing depression in adulthood. We know that being maltreated in childhood is increasing the risk for depression in adulthood. It's not a good thing to experience. But they also found that not everyone that experienced childhood maltreatment develops depression. Some people seem to be resilient. Why is that the case? Well, it might be because genetic factors moderate the effects of childhood maltreatment on depression. And this is exactly what they found. So they found that if you have two long versions of the serotonin transporter gene, you are no more likely to develop depression if you experience severe maltreatment than if you haven't experienced maltreatment, suggesting you're genetically resilient in the face of childhood maltreatment. But if you have one or two versions of the short ones, then we see an increased risk to develop major depression. If you have only the short versions of the genes, but you experience no maltreatment, you're no more likely to develop depression than the others. If you experience severe maltreatment and you have the long versions of the gene, you don't develop depression. In the absence of maltreatment, there's absolutely no differences between people that have the short version of the copy. So this finding suggests that the short version of the serotonin transporter gene increases the risk to develop depression, but only when having experienced a negative environment, right? So this was the interpretation of this finding, and the interpretation is consistent with the vulnerability stress or diathesis stress model, also the vulnerability model. According to this model, two individuals may not differ from each other on their level of functioning, that could be well-being, in the absence of a negative experience. It is the negative experience that triggers different responses. So we have a negative influence, then we have one individual that seems to be resilient even in the face of adversity, and another one develops problems. That individual would be considered vulnerable, and that could be due to a genetic factor. That individual seems to be more negatively affected by a negative experience. So that's viewed from a perspective of psychopathology, viewed from a perspective of vulnerability stress. It's the combination of vulnerability and stress that leads to a negative outcome. But more recently, I have been involved with Jay Belsky, who put forward a model of differential susceptibility, suggesting that maybe what we see here is not that some people are more vulnerable to negative things, but some people may be more sensitive across the board to what they experience. That means the resilient individuals maybe should be considered as those that have a lower sensitivity, and the vulnerable ones may just have a higher sensitivity. Now, if that's true, we would expect that in response to positive environments, that the low sensitive ones would show a lack of response to the positive, and the highly sensitive ones would show an increased positive response when experiencing positive environments. And genetic factors that are associated with both an increased response to the negative and an increased response to the positive, those genes would be considered sensitivity genes. Whereas other genes that are only associated with an increased negative response would be considered risk genes or vulnerability genes. They increase the risk to develop something that is bad. Whereas sensitivity genes, they just increase the sensitivity of an individual to both negative and positive experiences. So the study I just showed you beforehand, they suggested that in the absence of maltreatment, there was absolutely no difference regarding depression as a function of genetic differences, right? But they only contrasted the absence of maltreatment with severe maltreatment. Now, this is one of the replication studies. These findings have been replicated. In this study, however, they contrasted an adverse family environment with a supportive family environment in the prediction of depression. What they found, if you have the long version of the gene, it doesn't matter whether you experienced an adverse or a supportive family environment. It doesn't make a difference regarding your depression scores. So this is consistent with the previous finding. They also found that if you have two copies of the short gene, you're more likely to develop depression than the others if you experience an adverse family environment. That's also consistent with the previous study, which suggested that if you experience severe maltreatment and you have two copies of the short version of the serotonin transporter gene, you're more likely to develop depression. What they also found, which is not consistent with the previous study, is that if you have two versions of the short allele of that gene, but you experience a supportive family environment, that you actually have less depression than the others. So this is consistent with the model of differential susceptibility, or the idea that people differ in their general sensitivity to environmental influences. So this changed the story completely, because now it suggests that the short version of the serotonin transporter gene is not a risk gene for the development of depression, but it is a gene that increases the general sensitivity to environment. So people that have that gene, they also benefit more from positive experiences. This is a study, we looked at depression. This is a study looking at life satisfaction as a function of life events. And they were testing the same thing here, looking at whether the serotonin transporter gene is moderating the effect of life events, both negative and positive ones, on life satisfaction. What they found is consistent with the understanding that the serotonin transporter gene may increase the sensitivity to both negative and positive life events. Because they found for those that had two copies of the short version of the gene, they had lower life satisfaction if they experienced negative life events, but they had the highest life satisfaction of all the people if they experienced positive life events. But those that had two copies of the long allele, the long version of the gene, the resilient individuals, their life satisfaction was not affected at all by what they experienced, meaning they're not sensitive to what happens around them. They're not as sensitive to their experiences. So there's basically two sides to environmental sensitivity. Individuals that differ in their response to negative experiences, which is captured in the vulnerability model, the stress model. This is the predominant model in psychiatry or clinical psychology, looking at differences. Why are some people developing problems and others not? And then we have this positive end here that suggests that people differ in their response to positive experiences. That has not been investigated in great detail. Why? Because we didn't have a concept. We didn't have terminology to describe that some people are doing better when they experience a positive environment than others. So we published a paper a few years ago and suggested vantage sensitivity as a term to describe those individual differences in response to positive experiences. So that model suggests that two individuals may not differ from each other in the absence of a positive experience. But once we have a positive experience, a positive influence, some people are maybe more likely to benefit from that positive influence. They show vantage sensitivity as a function of vantage sensitivity factors. And other people, they may not respond at all. They show vantage resistance as a function of vantage resistance factors. So that's the positive end, the positive end of environmental sensitivity. And just one example of where we were looking at the positive effect of a resilience-promoting school-based intervention, and we were testing whether children that have a particular gene variant of the oxytocin receptor gene, whether they would be more likely to benefit from the intervention than others, and that's what we found. So this is a small study that has been, it's just for illustrative purposes, but this is a school-based intervention that took place in London, and we have pre, post, and a 6-month follow-up assessment. And some people have the C allele, and some people are homozygous for the T allele. And what you can see at the beginning of the intervention, there is no significant differences between these groups. So the differences, the genetic differences, didn't predict resilience differences at that stage. But over time of the intervention, it was only those that had two copies of the T version of the gene that increased in their resilience as a function of the intervention. So this is an example of vantage sensitivity, that certain gene variants seem to increase the probability of people to benefit from positive supportive components of the environment, like psychological intervention. This has been replicated in another sample with a different intervention. So we have to rethink genes here. I have to rush through this, but the traditional understanding is that genetic factors predict our behaviors, right? But what we have found out in recent years that the opposite can be true as well, that behavior can affect how genes work. Behaviors, an experience that we make, don't change our genes, but they predict how they function, and that's through epigenetic modification. So epigenetics is related to genetics, but it means changes in the phenotype, in the appearance or gene expression that are caused by mechanisms other than changes in the DNA. And there are several mechanisms, methylation and acetylation. And methylation means this is a methyl group. If that methyl group binds to specific areas of the DNA is being switched off. So the way epigenetics works is the DNA is exactly the same, but certain molecules bind to the DNA and then silence that DNA so that it can no longer produce proteins. And environmental influences have been found to affect methylation. So nutrition, stress, early experiences, trauma, all of those experiences, but also positive experiences, they have an impact on methylation of our genes. That means our experiences can predict which of our genes are active, which of our genes are passive. So I come to my conclusion. So we know that well-being has a specific heritable component based on the behavioral genetic studies, based on the molecular genetic studies. It's not the result of one particular well-being gene, but it's the result of many different genes, probably thousands or hundreds of thousands of different gene variants that each have a very tiny effect. Genes and environment, they interact with each other. The way they do that is through epigenetic mechanisms. So positive experience that you have may change the methylation pattern on your DNA, and that will predict which of your genes are active and which are passive. And all of that suggests that genetics or psychological well-being is highly complex. And that would be the end. Thank you very much.
Thank you, Michael, very much. That's fascinating. We've got a few moments for questions, and since this is the last one of term, after we finished at 2 o'clock, there will be an opportunity for those who want to hang around for a little bit longer to engage in discussion with the speaker. But first question, I can see Dennis has already got his hand firmly in the air. Who's got the mic? Oh, yeah, Eleanor.
Thank you very much. Is this on? It is on. Right. Thank you very much for a very clear talk. That's very helpful. I have a question just to relate your GWAS study with your candidate gene studies. So GWAS, as you rightly said, turns out that every possible candidate has a very, very small effect. Yet when it comes to these sort of candidate gene studies like, your serotonin transporter, the way that it's presented then is, well, this has a large effect, if you have SS or SL or L and so forth. So there seems to be, do you feel a tension between the GWAS study and that kind of interpretation? Because on the face of it, you might expect the variation to be very, very, very small from any single possible gene variant. So,
yes, there is a bit of a war happening between the people that do GWAS studies and those that do candidate gene studies. I think there are two different approaches, and we should use both, because one of them is hypothesis-driven, and the other one is hypothesis-free. Well, I actually found that the serotonin transporter gene is one of the few where we know a lot about it. We know it is functional, and we know that there are animal models, knockout mice, that don't have the serotonin transporter gene, and we can see behavioral differences for them. We know that it is associated with some brain activity, reactivity, so it's associated with the amygdala. If you have the short version of the gene, the amygdala is more reactive to negative stimuli. So we do know a lot, but it's a very different approach. So for the candidate genes, you need to have a biological hypothesis behind that, and many of them haven't been that helpful, or haven't been replicated. The GWAS approach helps you to mine the data to find some associations, and that will result in a new candidate gene. So then you have a gene variant that suggests is associated with well-being, then you can go and look at that specific gene variant. But many of those gene variants are found in genes where we have no understanding. We don't really know what that genetic difference does. So that's where things become more complicated, because it's fairly easy running a GWAS study and maybe identifying a few gene variants, but it's another thing then to take those gene variants and really understand how they contribute to a particular outcome. Also, another finding is that with a lot of the GWAS studies, those genes are not specific. So the genes associated also predict height to some degree. So, but it does, I mean, it does make sense. We have about 20 these genes. There's a process called splicing, so the same gene can result in different proteins. So it's a bit like the different letters. So by just looking at the letters, we don't really, it's just very complicated. Function of genes themselves, but then also in relation with the environment, to predict highly complex phenotypes like well-being or any other common behavioral traits in humans, it's very challenging. And I don't think we have the methodologies yet to do that.
Well, lots of hands going. Take a hand down here.
Yeah, sorry, you mentioned epigenetic modulation, is
Yeah.
that what you called it? Yeah. Okay, so it's in relation to that section, okay. It's about altruism, okay, as something that you could introduce that would change behaviour away from the more negative elements of behaviour. The reason I asked this, because I had a discussion with a scientist recently, and he said he was a geneticist, he just doesn't believe it exists at all.
Okay.
And I said to him, well, it might be nice if we imagine it, but I just wondered what your position was on that, and where it fits into in that section.
Altruism.
Yeah.
Okay, so we know that there are several studies suggesting that if you're altruistic, if you help other people, it actually increases your own well-being. So by doing good to other people, it increases your own well-being. That's I think a fairly consistent finding. There's strong research, experimental research, suggesting that this is the case. So it would make absolute sense to me that if you do good things for other people, that may have an impact on an epigenetic level, that it may change some of the gene expression that you have. So yes, there are genes. Yes, there is epigenetic modification. So much of the epigenetic mechanism actually are active during the prenatal period, during pregnancy, because they program the different cells. So all the cells have the same DNA, but it's the epigenetic signature in each of those cells that determine what the cell actually does. So people thought that all of the epigenetic mechanism, all of that is done once, after birth. Every cell knows what they have to do. They're programmed epigenetically. The more recent finding is that actually that process is still ongoing and is influenced by environmental factors. So nutrition has an impact on that. Physical exercise, if you go running, that has an impact on your genes, and it should have. It's the environment communicating with the genome, and the genome then knowing what proteins are being needed.
A very legitimate intervention.
Yes, yeah. So everything that we see, all our behaviors, every single thought that we have is influenced by genetic factors. But it's not that genes only play a role early on in development and they predict our personality and how we look and all of that, but the genes are constantly in interaction with the environment. The genes are constantly being read into proteins, and because we constantly have to adjust to the environment, that interplay is all the time ongoing. So if there's a positive influence, a positive environment, an intervention that has an impact on us, it will have an impact. It will be included in that complicated dynamic process between our genetic factors and the environment.
It's 2 o'clock, so I'm going to have to draw the questions to a close there. Otherwise, you're going to be late for your next appointment. That'll mean your stress levels will go out, your epigenetic modifications will change, and then you'll be unhappier. We've had a fascinating afternoon. Thank you ever so much for stimulating us to think, and there is an opportunity for those who want to stay for a little bit longer to discuss further. But thank you very much.
Well, thank you.